Hello and welcome back to part 2 of antimicrobial medications. Hope you're doing all right. So think about it. Resistance to something or susceptibility to something has to be determined. And so there's a couple ways to do that. So a real common way to do that in a laboratory is a conventional disk diffusion method. And so there are two men that came up with it, Kirby and Bauer. So it's kind of named after them. So it's called the Kirby-Bauer disk diffusion test. And so what you would do is you would take a plate and you would make a lawn of bacteria. So you would have a standard sample of whatever strain you're trying to test, and you'd spread them uniformly. And then there's discs of antibiotics, and they're sold in certain concentrations, and so you buy them. And so then you would place a disc onto the agar plate, and then you would incubate it. And so what's going to happen is the drugs are going to diffuse out, and so it sort of makes a concentration gradient. So closest to the disc is the highest concentration, and as it goes further away, it's a lower concentration. But what you're going to get if a bacteria is susceptible or sensitive to a particular antibiotic, you're going to get a zone of inhibition. So you're going to get this clear zone. And so you would measure the diameter of the zone, and you would have to compare it to a particular chart, because you have to think these are different size molecules and they're diffusing. So there is a chart. You can't just go, oh, well, this one, oops, my pointer's not working. This circle is bigger than this one, so you can't automatically say that this would be a better drug of choice. And so like I said, you have to look at a chart, so it takes into account molecular weight, stability, and amount. And so there we go. So this is a chart. And so these would be the different discs that you placed on it. And then two, it's sort of standardized for different strains of bacteria. But if the zone is small, then that would show resistance. You can also have intermediate sensitivity and then sensitive. So you would really want to look for something, choose an antibiotic that particular bacteria is sensitive to. And so that's one of the ways you can determine susceptibility to a particular antibiotic. Susceptibility and sensitivity, both those words mean the same thing. Another way, it can be done by serial dilution. So this can be done in test tubes, or it can also be done in a 96-well plate. But you can come up with what's called a minimum inhibitory concentration, abbreviated MIC. And so it's kind of what it sounds like. It is the smallest concentration of a drug or the highest dilution in this particular test that visibly inhibits growth in vitro, so in a lab. And so this is just kind of showing you, you would take tubes and you would then dilute out your drug. And so this is showing you a 1 to 2 serial dilution. So these are getting less and less concentrated. And so you would look for growth and see the minimum inhibitory concentration. See growth here, and then you don't see growth at the higher dosage. Same down here, you saw growth all the way up to 0.5 micrograms per mil, and then higher you do not see growth. And so this would be another way. What this is useful in determining is going to be that smallest effective dosage of a drug. And remember, that's kind of part of our therapeutic index. We want this to be small. And two, you can also, it gives you a comparative index against other antimicrobials. So you could test the same organism using different drugs. So both of those methods, they take time. They're somewhat labor intensive when you start looking at all those tubes and you start diluting things out. I just kind of want to know clinically there's going to be other ways to do it, or there's faster ways to do it. So the big thing, commercial modifications of susceptibility testing. There's something called the E-test, and that uses these strips instead of the Kirby-Bauer method. But once again, you're going to look for intersecting zones of inhibition. Some automated systems where you use little cards, put it in a machine that can visibly determine growth rates via turbidity, and then determine that minimum inhibitory concentration in like 6 to 15 hours. There's also new systems that can just detect the genes that encode antibiotic resistance. So there's just other faster ways of doing that. In a lab, we would use probably the Kirby-Bauer method. We could also do those serial dilutions to figure out the dosage. All right. So the big problem, increasing use and misuse of antibiotics is going to select for resistant microorganisms. And so this picture, it's showing you here you've got this population And so you see S stands for sensitive or susceptible, and R stands for resistant. So when an antimicrobial medication is added, those that are sensitive are going to die, and then only the ones that are resistant can grow. So that's what I mean by selecting. So just kind of like evolution, survival of the fittest, only those resistant are going to grow. And so over time, through our, not quite 100, I guess 80 years of using antibiotics, there are a lot of microbes that have resistance. If you think about it, there have to be resistance genes because it's microbes that are making these particular antibiotics. So if they're making an antibiotic, they naturally carry that gene, and we know that they share DNA. Okay, so looking here, just showing you the progression, only 3% of Staphylococcus aureus originally were resistant to penicillin G. Now about 90% of the strains are resistant. And like I said, it's growing at an alarming rate. We're going to look at some impact on cost, complications, outcomes of treatment. Dealing with these problems requires understandings of the mechanisms and spread of resistance. So of course we're going to look at that. So let's look at those mechanisms of antimicrobial resistance. So this is showing you resistant cells. You have an antibiotic, it's going to have a target, and then it's going to exert its action either through interrupting cell wall synthesis, protein synthesis, nucleic acid synthesis, the synthesis of folic acid, or that messing with that membrane integrity. So one is antibiotic inactivation. We kind of talked about this a little bit when we talked about the penicillins and the beta-lactams. Bacteria can produce enzymes that they secrete extracellularly that basically interfere with the drug. And so other ones, only one I didn't mention, chloramphenicol acetyltransferases is just another example. Other things that can happen is alterations of the target. So just minor structural changes can prevent binding. And so those proteins that are doing that cross-linking, they can have just a slight difference, and then those beta-lactams can't bind to those penicillin-binding proteins. Other things to decrease uptake. So if you don't take it in, then it can't hurt you. And so porin proteins prevent antibiotic entry into cells. So specifically in gram-negatives. Also to increase elimination. So those efflux pumps, if they are constantly pumping out that antibiotic, then that too would make it resistant. So increased production or structural changes of pumps allow faster removal. And so if they get that gene for that efflux pump, they'll eject that antibiotic. Okay, so there's two ways to gain antibiotic resistance. It can either be through spontaneous mutation or horizontal transfer. And we talked about this before, and I think we even watched a YouTube with that humongous petri dish and showed how those spontaneous mutants, under selective pressure, they outgrow those that are susceptible. Keep in mind, the antibiotics aren't causing these mutations. It's just selecting for those mutants in a populace that are no longer susceptible to the antibiotic. And so this is the regions a lot of diseases are now being treated by combination therapy. So 2 or more antimicrobial medications simultaneously prevents the growth of mutants that might be resistant to one of the antimicrobials. The odds of getting 2 random mutations at the same time that make you resistant to 2 or more antibiotics at the same time is really, really low. And so that's just why combination therapy is a good idea, in particular when strains are becoming more resistant. So that horizontal gene transfer, hopefully this is review for you from our microbial genetics. And so horizontal gene transfers, so just different members of bacteria, could be different strains, species, even to this point of genre. The most common way of giving resistance is through conjugation, and so through that sex pilus, so transfer of those R plasmids. So R stands for resistance. Often there's going to be multiple resistance genes on that same plasmid. Those R plasmids originate from one either through spontaneous mutation or microbes that naturally produce the antibiotic. So genes coding for enzymes that modify aminoglycosides likely originated from Streptomyces species that actually produce the antibiotic. And that's just kind of what I was saying before. They can't make something that would harm themselves, so they're always going to be resistant to it if they're the ones making that antibiotic. Other ways through transformation. Remember that was that uptake of naked DNA from the environment. So when cells lyse, they leave their DNA, and another microbe, if it's receptive, can then pick it up if they're competent. And then 2, we just talked about viral infections. Viruses are sloppy. They can accidentally package that gene and then transfer it to another microorganism. So just that You can either get spontaneous mutation or through horizontal gene transfer. Usually it's through that conjugation and transfer of R plasmids. That R stands for resistance. All right, so we're almost done. So examples of emerging resistance. There was a big United Nations conference. I believe the first one was in 2013. So this is a big deal. The CDC publishes antibiotic resistant threats in the United States, and they give them categories as urgent threat, serious threat, and concerning threat. And so, no, I don't expect you to know all this. There's a couple that I'm going to point out because it's a great review of some microbes we've talked about a lot. You're going to see them often in healthcare situations. So we're going to talk about those. We have talked about this one extensively. So urgent crisis. Clostridium difficile causes that antibiotic-associated diarrhea, and so a pseudomembranous colitis, so the really bad kind. It is considered an urgent threat because it's related to the use of antibiotics, meaning they are already resistant. So they have a lot of disease resistant. They're very hard to clear. Carbapenem resistant Enterobacteriaceae. I'm not going to ask you this particular one, but I'm kind of highlighting the fact that the majority of these you're going to see in a healthcare setting. So healthcare associated infections, because in those environments, that's where you have a lot of antibiotic use. And so those environments are selecting for these resistant microbes. We're going to talk about Neisseria gonorrhea. Yes, the cause of gonorrhea, which you cannot become immune to because of the antigenic variation. So, yep, hang on. I get it going.
Gonorrhea is a major public health concern in the United States. More than 800,000 new infections occur each year, but because many people don't have symptoms, fewer than half are detected and reported to CDC. Untreated gonorrhea can cause serious health problems. For women, it can increase their risk for a life-threatening ectopic pregnancy. And for men and women, the infection can cause conditions that can lead to infertility. It can also increase a person's risk of getting or giving HIV. Medication to treat gonorrhea has been around for decades, but the bacteria has grown resistant to nearly every drug ever used to treat it. In the 1980s, resistance to penicillin and tetracycline grows, and they are no longer recommended to treat gonorrhea. Fluoroquinolones are the leading drugs to treat gonorrhea in the 1990s, but the bacteria was adapting to the drugs. By the 2000s, resistance to fluoroquinolones steadily takes hold. CDC modifies treatment recommendations throughout much of the decade to keep pace. In 2000, the drug is no longer recommended to treat people infected in Asia or the Pacific Islands. By 2002, this recommendation extends to California. By 2004, CDC no longer recommends it for men who have sex with men in the United States. By 2007, resistance is so widespread that CDC no longer recommends fluoroquinolones to anyone in the U.S. to treat gonorrhea. Only one class of antibiotics, known as cephalosporins, remains to treat the infection. There are two main cephalosporins to treat gonorrhea, the oral drug Cefixime and the injection ceftriaxone. In 2010, CDC takes additional measures to combat resistance, recommending dual treatment with either Cefixime or an increased dose of ceftriaxone and azithromycin or doxycycline. But just 2 years later, in 2012, CDC updates treatment recommendations again in response to data suggesting the oral cephalosporin Cefixime is becoming less effective and gonorrhea has become harder and harder to treat. Today, we are down to one last recommended treatment option, dual treatment with an injection of ceftriaxone and an oral dose of azithromycin. Little now stands between us and untreatable gonorrhea. There are already troubling signs with the last recommended treatment. Abroad, infections resistant to ceftriaxone have been detected in several countries. And within the U.S., while not common, resistance to azithromycin has been found. The U.S. hasn't seen treatment failure when using these two drugs together, but drug resistance is rising, and the pipeline for new drugs is shrinking. Our last treatment option won't last forever. We must keep drug-resistant gonorrhea as a leading priority. Learn more at cdc.gov/std/gonorrhea/A.
Yeah, so that's a scary thought. Nobody wants antibiotic-resistant gonorrhea, so untreatable gonorrhea. So Clostridium difficile, Neisseria gonorrhea, they are on the urgent threat level for the CDC, meaning they are tracking just the strains throughout the world. Other things about the CDC, I mean, they really do a good job. They are definitely on it. Like I said, those drugs that attack the cell membrane, this is just a side note, they've identified bacteria now that are resistant to those polymyxins, and so the CDC are watching the spread of that resistance. Originated in China, and so they really are. They have a global footprint. I guess basically what I'm saying. All right, so the next threat level is serious. So those were urgent. So drug-resistant Campylobacter. And so we talked about Campylobacter. That is the #1 cause of acute diarrhea in the United States. And they are now increasing azithromycin resistance and also ciprofloxacin. And so that is going to be, that's on that serious threat level. These other ones, I'm just kind of pointing out, like healthcare-associated infections. These are all causes of healthcare-associated infections. Candida, so yeast infections that are resistant. Once again, Enterobacteriaceae, common microbe, so a normal member of our microbiota that in a hospital setting can cause disease. Vancomycin resistant as well. Once again, these are all healthcare associated infections. So just kind of pointing that out to you. Once again, it's not the antibiotic use that's causing the resistance, but it is selecting for microbes that are resistant. Pseudomonas aeruginosa. We've talked a lot about this microbe. It is difficult to kill. It is a common cause of many healthcare-associated infections, so pneumonia, cystic fibrosis patients is a biggie. Remember, it's also the #1 UTIs in hospitals, big and wound infections, fatal sepsis, pneumonia. So it's just a really bad one. And so it has multi-drug resistance, so it is on that serious threat level. Okay, concerning. So these are just concerning. These aren't, they're not serious, not urgent, so we're dropping down a little bit. Once again, so we've talked about methicillin-resistant Staphylococcus aureus. Once again, healthcare-associated, big in healthcare, skin and wound infections. And so they are now methicillin-resistant, and so they're going to only be treated by vancomycin. But the problem is that now we're also getting some vancomycin-resistant Staphylococcus aureus. And so just kind of both of those, because they're being selected for in those hospital environments. And then tuberculosis, your book really covered that one quite a bit. Mycobacterium tuberculosis, it is not endemic in the United States, but is still endemic in other countries. And it is difficult to treat anyway because it's a mycobacterium. Not many antimicrobials work against it. It also grows really, really slowly, so it's kind of a pain to treat anyway. So really long treatment care, and now there's just different levels of drug resistance. So that one is always a concern. So let's look at the 4 ways to slow the spread of antimicrobial resistance. The big thing is responsibility of physicians and other healthcare workers. Increase efforts to identify the cause of an infection. Stop prescribing antibiotics for viral infections. So only prescribe them when appropriate. So your book gives you this, and it's funny because I actually listened to a podcast about just the alarming rates of antimicrobial resistance to the fact that they expect by 2050 more people will die of infections from these resistant bacteria than will die of cancer. But this is just showing you the rates of antimicrobial prescriptions per 1,000 people, and you see that it's much higher in certain states than others. So you can't say, oh, well, these are all the sickly states. So it's probably just, due to that irresponsibility, and they're being overprescribed in those areas. Other thing, responsibility of patients. And so there's something you're really going to have to pound home and encourage with your patients. Carefully follow the instructions, even if they're inconvenient. Take all of your medicines. Take them at the time you're supposed to. So if you skip a dose, you know that we're going to drop below that minimum dosage that's required for killing. 2, when people feel better, they stop taking their medicine. So failure to complete treatment may not kill the least sensitive organisms and allow subsequent spread. Educating the public. One of the reasons I think that antibiotics are overprescribed, someone goes to the doctor, they have a respiratory infection, it is more than likely a virus, but they're pretty angry and kind of pissed off when their doctor goes, yeah, you have a virus, and so the doctor just sort of writes a prescription anyway. Well, if it doesn't clear up in a week, here, take these, and of course people immediately go home and take them. Big thing, antibiotics are ineffective against viruses. And so that misuse, once again, you're just selecting for antibiotic resistant bacteria. And 2, the more you take antibiotics, your normal microflora will also become antibiotic resistant. And then when you do get a pathogen, they can transfer that R plasmid to that pathogen. It really is going to have to be a worldwide effort. There are many countries in which antibiotics are just sold over the counter without a prescription. So anyone can just go get that, actually Latin America and Asia. Other things too, the use of antibiotics in animal feeds. I believe it was around the 1950s, maybe it was a little earlier. They figured out that if you gave a low therapeutic dose of antibiotics to animals, that they would grow bigger faster. And so pretty much throughout the world, they said, hey, this is a great idea. Pretty soon, though, as early as the 1970s, there were scientists like just going, hey, wait, stop this, because those antibiotic resistant was spreading into normal microflora and just throughout the population. And so, yeah, really need to stop that. It's been banned in Europe. The U.S. also has some regulations that now antibiotics should only be used if to treat an animal. And so antibiotics are no longer in feed. It requires a veterinary prescription. So we are really working to reduce the amount of antibiotics used in raising animals, because it has been linked to antibiotic-resistant salmonella, because we know that we have a lot of these microbes not only grow on us and cause disease, they grow in animals. So, yep. All right, whoo-wee. Okay, so we're just going to kind of run through, I kind of gave this in brief as well. So antibiotics don't work on viruses, and so antiviral medications work on viruses. The problem with viruses is they grow inside host cells, so something that's selectively toxic is difficult to find, because once again, they don't have cell walls, they don't have ribosomes, they don't have other structures like bacteria do. So the big thing is these drugs are going to focus on preventing replication. And I did kind of veer from your book. Once again, your book got super heavy. So the big things, if you kind of remember, are stages for viral infections: so attachment, entry, synthesis, assembly, and release. So the 3 modes of action are just going to be barring entry, so either barring penetration or encoding. Then you remember they have to replicate, so we can block transcription or translation. And so there's drugs called base analogs, so AZT, to treat AIDS. Acyclovir is another one. Instead of when it goes to build its genetic material, there's going to be an analog that basically stops that production, or reverse transcriptase also. So just kind of puts a base that you can't build upon. So also, or another drug can prevent maturation of viral particles. We talked about protease inhibitors. Remember, viruses, they make their big long protein chain and then they have to cut it up to make their pieces parts. So there's some of those antiviral drugs that inhibit that, particularly when we're talking about influenza. So neuraminidase inhibitors are going to block that, so then that virus particle can't leave. So just sort of blocking our stages in viral replication. There's not a lot of antiviral drugs out there. You've probably heard that on the news. And so there's really only certain diseases that have antiviral drugs that are actually effective. So clearly HIV, hepatitis B and C, herpes viruses, so yes, treating of genital herpes, varicella zoster virus, cytomegalovirus, Epstein-Barr virus, so those. There's Tamiflu for influenza, not quite as effective. Once again, Big Pharma is going to focus on those chronic diseases, because if someone's on these drugs, they're going to take them for a really long time. So that's why there's more antiviral drugs for those particular viruses. All right, so why is it so difficult with antiviral drugs? And this will kind of give you a better rundown.
Viruses are difficult targets for selective treatment because they rely to a large extent on the host cell's metabolic activities for their replication. Most antiviral agents inhibit viral uncoating, interfere with viral nucleic acid biosynthesis, or prevent the assembly and release of viral particles. After the virus enters a host cell, the protein coat must be removed from the nucleic acid in order for replication of the nucleic acid to occur. Amantadine blocks penetration and uncoating of the influenza A virus. Phoscarnit is an analog of pyrophosphate. It binds to the active site of DNA polymerase and blocks the cleavage of pyrophosphates from nucleotides. It is used to treat cytomegalovirus and other herpes virus infections. Replication of nucleic acids involves additions of nucleotides to the free 3' end of the growing chain. Nucleoside analogues are incorporated into nucleic acids during synthesis, but then prevent further synthesis. For example, azithothymidine, AZT, an analog of thymidine, becomes phosphorylated in the cell and is then incorporated into nucleic acid by reverse transcriptase. Since the 3' position of the deoxyribose is occupied by nitrogen instead of a 3' OH, no additional nucleotides can be added. AZT has been used in treatment of retroviral infections. The HIV genome is transcribed into messenger RNA and then translated into a large polypeptide. A site-specific protease cleaves the polypeptide into functional proteins. Protease inhibitors interfere with this protease and prevent production of functional proteins. Protease inhibitors have been used to treat AIDS patients. Some interferons are antiviral. These proteins are produced by an infected host cell. The secreted interferon binds to adjacent cells of the same kind and stimulates production of enzymes that degrade messenger RNA and inhibit protein synthesis in these adjacent cells. Interferon has been used to treat Kaposi's sarcoma in AIDS patients, chronic hepatitis B, and hepatitis C infections.
Well, there you go. Hopefully that made it clearer for you. And yes, so interferon, that was part of that innate response. And so that too can also be used therapeutically. Okay, so fungal drugs. So yeah, so fungi are eukaryotic. So once again, they're going to be really difficult to target because they closely resemble human cells, so more close than bacteria. And so antibiotics are generally going to be ineffective against fungal infections, and so there really aren't that many. So there's very few for systemic use. Most of the antifungal drugs are going to be topical, so used for those dermatophytes that we talked about. So think about, ringworm as an example. So what fungus do have that's different is they have the steroid ergosterol. And so we don't have that. And so most antifungal drugs are going to target the synthesis of that particular steroid since we don't use it. And so as you can see, it's just kind of showing the majority of those antifungal drugs. And no, I'm not going to ask you any of the names of these. We had talked, I believe, about griseofulvin when we studied fungi. That's just a common one for treating, like I said, those dermatophytes, so, jock itch, athlete's foot, those sort of things. All right, so let's talk about treating protists. Remember, protists, there's just a wide variety of protozoans. So when we get to like the protists and the helminths, there's not a lot of drugs. We'll talk about the reasons why on the next slide. But malaria is a big thing, and it's been around for a very long time, and it affects a large number of people. And so traditionally, it was the quinine was the go-to drug. But of course now it's quinine and all of its relatives, so quinolones, chloroquine, or primaquine. And so remember we talked about Plasmodium vivax, but there's different species of Plasmodium. The other things too, remember it has a complex life cycle. And then two, another problem in treating malaria, that in certain regions, some of the plasmodiums have become drug resistant. There is a new drug out, artemisinin, there you go, and that is now being used in combination with quinine derivatives. And so that's had really good results, and the woman that discovered it also won a Nobel Prize. So metronidazole, also known as Flagyl, is actually an anti-amoebic, so it is effective against that amoebic dysentery, so Entamoeba histolytica. Flagyl, like flagella, it is also effective against Giardia lamblia, so also causes diarrhea, gastrointestinal tract disease, and then Trichomonas vaginalis, we talked about that as an STD or STI. And so just those are just some common anti-protozoan drugs. All right, so problems with helminths, so parasites. There's really relatively little research and development being done. This is because most parasitic diseases, and also for the most part protozoan diseases, are concentrated in poor areas of the world, where they can't afford the medications anyway. Once again, it's going to come down to money. Most chemicals for those helminths, because think about too, helminths are so large, really difficult to find drugs that treat them. They're going to interfere with those neuromuscular functioning of worms. So they're either going to somehow paralyze them or give them muscular contractions, but either way, so they can't move. So some examples: mebendazole, ivermectin, praziquantel. And so those are just sort of three of the really common ones. If you give your dog a heartworm medicine, that too is ivermectin. So yep, cross species. And so just remember the helminths, we've got our nematodes, so we have our roundworms, we've got our tapeworms, and then we have flukes, so cestodes and trematodes. So you know what this means, gang? Yes, this was it. This is it for lecture. Have an awesome day.